Time for our morning plenary, and I have the honor of introducing today's speaker, Dr. Michael Proctor. Some quick background about Michael. He received his Bachelor of Science from the University of British Columbia in 1995 and his PhD from the University of Calgary in 2003. He then went on to do a postdoc with Dr. Mark Boyce at the University of Alberta from 2003 to 2004. Michael has researched grizzly bear ecology and conservation since 1995, and today he is an independent research scientist and currently the lead Canadian researcher for the Trans-Border Grizzly Bear Project in southern British Columbia. He and his colleagues utilize landscape genetics, GPS telemetry, and ecological monitoring, and he specializes in solving conservation problems for small threatened populations, identifying conservation solutions, and actually trying to implement them. Although Michael's happy as a research scientist, he is really a boat builder, a musical instrument builder, and a 5-string banjo player, all of which he forgot to bring today, but maybe next time. He's also a mean bluegrass guitar flat picker, so again, next time he'll bring that. Michael did his first mark recapture experiment at the age of 10, when he marked turtles with a dab of nail polish. His capture technique was by stealth and fast underwater swimming. Michael is all grown up now, kind of, and his talk today is entitled, Providing Adaptive Options for Grizzly Bears to Respond to Climate Change or Defragmenting the West. Will you please welcome Dr. Michael Proctor.
What he meant to say was I was the world's oldest grad student. My advice to all you students, I know that Mike Soleil gave you some good advice yesterday. I gave you some other advice, is stay in school and get your PhD a little earlier than 50, you know. I got to go back a little bit there. This is actually, well anyway, first of all, thanks for dragging me down to Missoula because Corey, my wife, and I was here like to, louder, I'm getting the wife signal, louder, okay. It's nice to have a constant critic in the crowd there, you know. Bring your wife to a talk, and you have to button up your shirt. But anyway, it's nice to get down to Missoula. I'm an ex-American. I drifted north in 1985. Last time I got hauled over at the border in every other couple of minutes when I'm crossing the border somewhere, they're always asking me, why'd you move to Canada? And I said, well, you're dreaming, pal. I never really follow orders that closely, but I always just say, you know, there's more big game animals than there are humans up there, and that usually ends the interview, and they say, okay, I understand. You know, it's pretty cool. So Canada's a pretty nice place. So this talk really is 2 talks. This is actually the punchline of the talk. This is the second talk. The first talk, I'm not going to tell you what it is. You're just going to see that, and then we'll revisit this talk. But the end result is. The first talk really is about describing the problem. It's fragmentation. It's a satellite view of North America looking at grizzly bears with a pair of binoculars. That is, we're looking at large-scale fragmentation from really high up. There's like 1 million square kilometers in the study, and there's 4,000 bears in it, and then we zero in and do an individual-based analysis. We're following the fates of 4,000 bears and finding the patterns from that. And then, once we got done with that, we realized we should try to go out and solve that problem, that fragmentation. So the other half of the story is the work we did trying to sort of defragment the West. And the West I defined from Missoula to Revelstoke, BC. That is the West, isn't it? It's kind of... oh, yeah, I was going to time myself. At any rate, this is a plenary, and I have never done a plenary, so I've thought I better be a little smarter. And I was a little intimidated to go up against Michael Soleil, and I really want to thank him. I know he's not here, for doing an excellent job of introducing the idea of connectivity. I don't think I need to convince you guys that connectivity is and fragmentation important issues. That's the best introduction I've ever had, because that's what this is about. But also, I think there'll be a lot of students here. We're at a university. So I had some themes that aren't really about the talk, but they're about little subtext that you should think about. They're hopefully interesting. And if they're not interesting, hopefully they're a little entertaining. But at any rate, the obvious one is fragmentation is really the issue of our times. It's really affects biodiversity, and especially with the overlay of climate change. But also, I want you to think in terms of meta-analyses. You know, there's a lot of little postage stamp projects in the world, but we can really learn a lot of more things. Bruce McClellan's the guy who taught me this, by bringing all those smaller projects together and looking at them in a whole, and you'll see an example of that. And also, to do meta-analyses like that, you really need broad-scale cooperation. It's really necessary to deal with the big problems we have. And what that means, so the cooperation can be done within the people that we agree with, right? Like here today, everybody, we all kind of agree on this. So you really need to get to cooperate with your colleagues, but you also need to cooperate out there in the larger world with the people who don't agree with you and that don't agree with each other. And it's very important to bring the loggers and the miners and the farmers and the conservationists and the hunters into the same room and make them part of this. And that's kind of a theme of this. And then this is sort of something a little, you know, this is just a reflective look back at us. You know, cooperate more, maybe criticize less. You just see a lot of infighting, even among the people we all agree with, and I just think we need to cooperate a little bit. And then last, well, maybe it isn't last, but explore with your analyses. You're going to see some exploration things. We're driven by hypotheses testing, but you'll see sometimes you got to just set that aside and go out and figure some stuff out. And I wasn't sure what the tone of this conference was going to be. I know there was some discussions about the role of science and conservation. Is it too late or whatever? But we're finding that really solid science can be a great agent of action. So, and travel a lot. It's I do a few projects around the world, and I think there's an Ecuadorian here. I worked with her and there. What I learned from the Ecuadorians helping on Andean bear projects, they'll put let's say they got a $40,000 science project to study bears. They also have a $40,000 social science parallel project to integrate all those solutions. That's an unbelievably impressive idea. And the more you go around the world, you start learning a lot of stuff like that. And here I was in the Galapagos. I ran into Charlie Darwin, and we had a hell of a conversation. It was totally fun. You know, things happen. I'm not sure what they put in that coffee I had that morning, but I learned a lot of stuff from Charlie. So the first half of this talk is this is the fragmentation side. This is the problem, and it's going to be a wildlife monograph. It's going to be mailed out to Wildlife Society members on January 1st. I'm going to be like one of those new mothers who has the first baby of the year. It's like a birthing. Get this damn thing out of my computer. But it's got 19 authors on it, and it's got 4,000 bears in it. And this is where we looked at fragmentation by looking at the individual movements of 4,000 bears across Western North America. And I'm going to, kind of whip through it here. You're not going to get a lot of details. There's it's four separate papers in one. So we're just going to, it's like water skiing behind a really fast boat today. So grab onto that rope there. This is a sort of the study area. I'm just going to show you a little bit about cooperation and meta-analyses. This is the distribution of bears in North America, obviously, the shaded area, and those are kind of the road networks. And this is what data has gone into this monograph. You're going to see it started out with the Yellowstone radio telemetry studies, Chuck Schwartz, Mark Harrelson. I'm not going to spend a lot of time on these guys. You're just going to flip through them. These are the guys who did the early telemetry studies. All their data is in this study, all these guys who did this work. You've seen all these names, Mike Jabot and Lana and Gordon Stanhouse got a little carried away in Alberta. And John Waller, and then even me, I got into the act here. But then we switched over and we started doing population surveys in 1995, and we've been doing them across North America since. Those are the DNA surveys, the hair grab. And these are what we did the first one there in 95. It was actually my directed study at UBC, the first silly little DNA survey. It's sort of taken over the world a little bit, and what's really cool is to see it spread around to different other species. So it's kind of been a satisfying time to watch that go. It was not my idea. I like to go on record as saying that. I was the first guy who did it, but it was Bruce McClellan's idea, so he gets a little gold star by his name. But you can see we kind of headed around British Columbia doing them. It kind of took off, and we just did more and more DNA surveys. And I think John Weaver did one right there. He's down there. And Kate Kendall did the mother of all, the U.S. mother of all DNA surveys down in the NCD, did a very good job. And then Stenhouse got carried away in Alberta again. And Clayton Apps really got carried away. But these are the telemetry projects and the DNA projects that make up the monograph. So it's a pretty big, this is the power of a meta-analysis. We can learn all this. And I had to sucker all these guys into trusting me with that data. That was the hard part. My Scotch bill is really expensive. But at any rate, that's the monograph study area, so I'll get into it. And just to back up just a tiny bit, just a quick little reference of the importance of fragmentation. This is the gray is the past distribution, historic distribution of bears. The blue is the current. Back in 1920, you might have all seen this map. That's what the grizzly bear distribution looked like. A bunch of little fragments poking around North America. This is the Texas view of North America on the left here. You know, that's the best joke I do, and I always have to point it out. But at any rate, back when Bush was the president, it went over a little better. But at any rate, you can see by 1970, 50 years later, those were all gone. So those small fragmented populations, they have a real challenging time making it. Of course, bears were under kind of a persecution paradigm at that point, and that's changed now. We all look at bears as a real positive thing on the landscape. But what I'm here to say is we still need to keep track of that fragmentation. Even though we like bears, we still got to look at that. So, this my only method slides. It took me a long time to make this video. I worked for weeks doing this. Oh, I'm sorry, Jeff. Actually, Jeff Stats made that video. And it doesn't always go the way you claim. But this is just a standard DNA method. There's some stink in the middle of that, and the bear goes in there sometimes if you got too many doggone big bears down here in Montana. They don't always cooperate. But what's really want to point out about this slide is we use this for population estimation. It's really a powerful tool. But what's really more important, population estimation, is how many bears are out there. It's really not a very interesting question. Who the hell cares, really? In a lot of ways, it's kind of important. But from an ecological standpoint, what's more important is to understand why bears are there and why they might be less or why they might be more. So we use this to study fragmentation, these DNA things. We also do it to habitat modeling, and we use it to ask questions on why. So I just want people to think well beyond population estimation. You'll see a little bit of that today. I've also used radio telemetry. I got about 800 radio telemetry bears in here. I didn't collar all those. I'm not that old. But most of the people you saw earlier did that. So anyway, we used a measure of exploration and hypothesis testing. And I know the profs here want to get you guys to test hypotheses, and that's good. But I'd like you to just think about, what was Darwin's hypothesis? It really wasn't. He just went around the world and tried to come to some understanding, and I'm sure he developed some. But it's nice to think outside your little science box. And so our first exploration was a look at things here. I'm not going to tell you what the method is. You'll see it in a little while later. I like to mix things up. This isn't methods, results, and everything. You're going to have to kind of think outside your box. So this is 3,100 grizzly bears sampled with DNA, right, across North America from Yellowstone all the way up to the Yukon. And I'd like to have a plug for Wildlife Genetics International. I think Jenna's around here, one of the employees, and did a lot of the work. I think they probably did the genetics on 3,099 of them. They did quite a bit of that work. Very excellent lab. But this is a genetic analysis that relies not on any population genetics, really no allele frequencies or no sample size issues. It just clusters bears based on their allele sharing. I'll go through it a little bit later. But basically, you can see just kind of there's this concordance, this geographic concordance. The bears are color-coded by north-south in that long tower. And then there's two little sidebars over here that don't really fit that mold. And you can see where they correlate to down there. And that just correlates to Highway 3 in British Columbia. It's just as simple as that. If you look across North America with that bear thing, it's just, we didn't have a hypothesis. We just threw that data in the thing and spit it out, and we go, wow, look at the difference there. It just helped us kind of understand what's going on at a big scale. You'll see a little more detail on it later. So we developed our first hypothesis, which was we're looking for patterns of gene flow isolation by distance. We know up north that there's a lot of isolation by distance. Dream on, man. We know that up north bears, their gene patterns, the gene flow follows an isolation by distance pattern. So it's just a simple cartoon there. As geographic distance goes up, with bears, genetic distance goes up. It's a very simple idea. So we hypothesized that isolation by distance would probably explain a lot of the variation up north, and in the southern area where it's a little more crowded with humans, that it would be less important. That's the basic hypothesis. Our analytical method, and you can't run a standard regression with genetic data. You have to run a Mantel regression that compensates for the autocorrelation. If you really want to know how that works, you just ask my wife. She's sitting there in the blonde hair over there. And so what we found is, very simply, that isolation by distance explained 38% of the variation in genetic distance between those population center, or those clustered samplings that you see on those black dots. And when we started including ice fields, it rose to about 55%. And then down in the south, it explained about 10%. So we knew we had something going on, that isolation by distance wasn't operating at a very large extent in the southern half of the distribution of bears. So this is just sort of a first cut. We wanted to compare the north to the south and see what we get at this coarse scale. So our second hypothesis really concerned the southern section, and that hypothesis was there was anthropogenic fragmentation occurring. We wanted to test that. So our second exploration, this is where, again, and I'm going to explain that method a little bit, and I call it an exploration because we're just going to, well, anyway, it was can we determine the fragmentation with no a priori assumptions of clustering. A lot of these population genetics things, you got to go out there with preconceived notions, set up allele frequencies, make false barriers, and test them whether they're fractures or not. But what this does, it gets rid of us. We become a little more irrelevant. Irrelevance is underrated, you guys. So it's a nice place to be because we're going to let the bears tell us where those fragments are. So it really is an exploration. It's nice just to admit that up front. So we use this method, genetics. It's a program, and it's a very complex program to explain, and I will try just a tiny little bit because it's pretty powerful. Like I said, it's got no assumptions of group membership and no preconceptions required, and it doesn't use allele frequencies. You can throw 3 bears in there and it doesn't represent a population at all, and it'll place them, relative to the other bears. It's pretty powerful. And we have some pretty amazing examples of that, but we're not going to see them. But what it does is really kind of cool. I call it the sea urchin explanation. Every allele, it uses allele sharing, but every allele has a dimension. So it's a multi-dimensional thing. So you can just imagine a dimension. There's 2 dimensions on that graph. Just imagine 100 dimensions, right? So you got a million lines going everywhere. And if you forced them into a point of 0, it'd be like a sea urchin. You'd have a dimension going out from the center. But of course, it's not forced to 0. So you've just got all these dimensions. And it does, basically, it's doing allele sharing. So what we found a kind of a continuum of fragmentation across this southern landscape. We had, that was about, well, I'm not sure what the square footage of that, but we found areas that were very highly fragmented. You'll see examples of them. We found areas that were partially fragmented. We can see the process of fragmentation occurring halfway along, and we found places that were very connected and not fragmented at all. So we got the full spectrum, and this is one of the, when I say do meta-analyses, what you want to do is put together your study in a way that you have variation on the things you're trying to learn about, and that's where you get your inference, your power from that. So it was very important. We went to a lot of trouble to get places that were highly fragmented and places that were not, and that's where we came out. So this is what highly fragmented areas look like. This is some of the science of the obvious. I specialize in that. This is Yellowstone National Park. There's 150 kilometers of no bears in between. It's been isolated for several decades. It's obviously going to be fragmented, but that's what its signal looks like in that. Up here is the southern Selkirks, and just across that Highway 3, that's that one of those that earlier graph, and you can see this, those not a lot of sex happening across that highway, except we had a little guy there, probably a migrant. And this is areas where it's starting to happen, where we have partially fragmented. We have these clusters, and they're pulling apart, if you will, but they're not totally separate like that. And here, you can see the cluster; they sort of overlap, but those are obviously clusters. I'm suggesting that they're on their way to being fragmented, and that's across the cabinets. Here's the cabinet yak in the across the border, and then this would be the NCDE to one side, so it's across the Kookanusa Reservoir. And here's one across the highway in Alberta, where they're starting to happen. Not fragmented. This is across the Continental Divide. You might ask yourself, does a big Continental Divide fragment bears? Absolutely not. That's just one big happy. It kind of reminds you of a university campus and the amount of interbreeding that's going on. And this is across the Flathead River. This is a very large, I told you about it. There was a little, this is a very large valley with one of the biggest rivers, the North Fork of the Flathead. And, it was sort of a really good control because it's a huge valley with a big river. It's also like a very open breeding across there. There's no separation. There's a lot of gene flow. So at any rate, here's just a really cool example to show you all in one pot. It's kind of neat. So this is the BC North area. Here's Alberta, BC border. drift you guys up above the 49th parallel, and this is Alberta and British Columbia. And the X's are this area in red right there. Okay? And right across the border in Alberta are the little circles, and they're just overlapped each other perfectly. So those bears are running back and forth and having sex across that border, just like anybody. So now we're going to go south of the Highway 16, and there's a bit of an overlap there. I've drawn that oval a little bit, and those bears are obviously not in that same group, and that's across that highway right there, Highway 16. Now we go across the next highway, and we've got it separated out. So you can see it all happening at once. You can see areas where bears go run across their highway together, and then you can see where they're separated. So we did this, everywhere, and found that you'll see a synthesis of all this. I'm just going through the individual analyses types, but so we could let the bears tell us. So that hypothesis turned out to be true. We could let the bears find these fragments for us by just throwing in data all over the world or all over our study area, and we found that exploration worked pretty well. So our correlator, our second hypothesis, was that females might be fragmented more than males, and this is an important point. A lot of genetic analyses, if you're using genetics to understand your fragmentation, you don't have a way to separate the sexes. Males, little fancy, cute little sperm packets they can run genes back and forth across borders and all the time. But if females aren't going across those borders, you're having a differential influence. It's female fragmented rather than male. So it's the individual analysis, not the group analysis, that allows you to separate the sexes in your inference. It's a pretty important little distinction. And of course, we hypothesized that maybe the anthropogenic causes are happening. So here, we use several methods for this. We use the genetic assignment. We look for individual specific migrants. And here's an example of it. If you can see it, right there is a migrant in that population. This is a red dot, obviously originated from this population, but we caught it in the next one. It's a pretty obvious example of a migrant. We've got a lot of math to separate out putative migrants, which is maybe migrants, to real migrants. You can read the paper for that. But then in several places, we had to use radiotelemetry because there's a lot of places like this, you can't pick out migrants in there because the genetics are identical across whatever your potential fracture is. And that's where I use radiotelemetry to estimate those. And in a minute, I'm going to show you the results. We calibrated that. We tested in many places genetic inference for migrants and telemetry instruments for migrants. And then in many places, we did both of them together and compared our results to test out. It's nice to do these inferential genetic examples, but it's also nice just to nail it down with radiotelemetry. So to sort of remove the fear of believing the genetics, because it's hard to deny a radio collar bear who moves between two areas, right? And then we compared those. So here, just an example. That bear moved between geographic areas. Pretty simple. Like I said, I specialize in science of the obvious. So anyway, our result is we found plenty of movements between areas. We found a lot of females moving between areas, and we found a lot of males moving between areas. But the patterns were, whenever there's a large highway and some settled areas, we found minimal female movement, and we found reduced male movement. Okay, so we have, you're going to see some of the details. So we went in and itemized and quantified the movement rates of males moving between two adjacent areas and females moving between adjacent areas, and developed a migration rate between all these areas. So I was telling you earlier, we compared these results. The migrant rate was 0.023 on average across all our, I think we did 24 population pairs, and the migration rate was 0.31, a little different, but it was statistically not different. So on average, they detected a similar amount of movements. So our third hypothesis, way more interesting, is the why. I've just been describing what happened. Now I'm going to try to get into the why. And it's always interesting to take your study to that next level. And we're going to I put causes in there because these are correlative. This is not an experiment. These are correlative inferences, and they're not perfect. But we were checking out if bear movements were reduced by traffic settlement and human-caused mortality. We're just going to ask that question. And we used the migrant rates from that previous analysis. We've got female migrant rates between 24 population pairs and male migrant rates between 24 population pairs. And we quantified traffic settlement and mortality between all those. And this is the study area we use for that. The darker the red, the more human settlement there is. And embedded in there are the traffic volumes. And we computed the mortality rate. So I just said we used the mortality rates, and we used ANCOVA and linear regression because we had discrete variable. If some of you modelers in will know that, I'm not going to explain it in detail. But we used those variables between 24 areas. I think I've already said all that. We tested our models in the AIC for model selection using a small sample size. So our results were really pretty amazing, and we were kind of fascinated by it. And I had a bunch of text there to tell you what it is, but I'm just going to show you what it was. You'll be able to understand it. So I'm modeling a multivariate model here, and it doesn't come out real simply on a graph like this. So you just kind of bear with me. This is a good depiction of what happened, and you'll just have to use your imagination if you're too mathematically inclined. So this is movement rate on here. We got settlement on here, and this is a very disturbed, 100% settlement, and this is like 20% settlement, 50% settlement. We got males and females. So in the undisturbed areas, less than 20% settlement, we had as traffic increased. Remember, this is multivariate. As traffic increased, both males and females moved less between there. We found fewer movements as traffic increased, but males had more movements than females, but they decreased about the same. Parallel curves for your regression types. And when we got into the moderately settled areas, 20 to 50%, there was an abrupt drop in the female movement rate. It just plummeted almost down to 0. And the males, as traffic increased, it slowly decreased as well as a function of traffic in a univariate level. And as traffic, in disturbed areas, they both decreased as traffic increased. This is called a piecewise model. It's 3 separate linear regressions, or a broken stick, and it's useful whenever you've got abrupt changes. Here's what it would look like if we didn't use piecewise. That's just a pure linear regression. It shows you the same pattern, but it's not near as interesting because you don't find some of the subtleties in there. So we were kind of excited to find some thresholds where things change. This also supports the hypothesis that females were affected by fragmentation differently than males. It had a much bigger effect on them. And then now we're going to real quickly just look at settlement in its relation to mortality, and this is even a little more fascinating. So here, they both started out about the same, and as movement rate increased, mortality increased. So what the hell's happened there? What's happening there is the more you move, the more you die. There's a cost to movement. Should be a sort of a typical ecological precept. So we're sort of interested to find that. That's sort of the natural world. That's what you might expect. If you're going to disperse or go to new areas, there's sort of a cost to that ecologically, and it comes out in higher mortality. But then in the moderately disturbed areas, there was no difference. We couldn't find a trend in either direction. And then in disturbed areas, highly settled areas, as mortality increased, both males and females moved less. So we had a combo. So we had these complex relationships between mortality, traffic, and settlement, and pretty interesting. So the fragmentation synthesis, we're going to put all those results together into one map. And this is my next piece of advice to students. If you can't put your thesis or whatever it is into one picture, it's too complicated. I try to specialize in boiling it down. This is my $10 million map. It's an anthropogenic, female fragmented, non-equilibrium metapopulation. And what the hell is that? I don't know. I'm going to have to bring up some expert to tell us. But what that means is I found that differential female fragmentation. That means that the males are moving between places. It's being reduced, but what's happening is the females are not moving. So it's very important. Remember the guy that did the talk yesterday and he described genetic and demographic connectivity? He was the river otter guy. That was a very good point. We're talking about demographic fragmentation here because the females are fragmented so much. You little sperm packets out there, if you don't get there, there's always another eager little sperm packet behind you to take over your case. But if you can't move a female to get over there to augment a population, it's not near as important. So that female component is a very important part of this work. So here's what that looks like. This is this satellite view from the Yellowstone to halfway up Alberta. Those yellow dotted lines are the places where we found fractures in those clusterings. Remember the earlier clustering analysis? And then we overlaid the number of migrants between there. And the numbers within there, because they used the DNA surveys to gather a lot of this data, we have real data-driven population estimates for the whole friggin' female metapopulation. It's kind of a powerful map. We'll go through it in a little more detail later, but it ends up showing you the fragments. There's 571 bears. I think some of these are up now. there's about 1,000 bears here in this in the Canadian. These are the real population boundaries. Doesn't care what country you're in or what jurisdiction. And there's some small populations, and it gets up. And where we don't have data, I put in unknown. This is a data-based map. At any rate, some of the implications of this are kind of cool. What I think we have going on here is a spatial pattern reveals a temporal pattern. And I'll show you what you mean. But I think we started out with a panmixic population 200 years ago. We've got partial fragment. This is time equals space here. So panmixia was the past and also the north. And the partial fragmentation, as we go south, we get into that fragmentation. And then we get extensive fragmentation in the south. And this is what the distribution looked like several hundred years ago, the dark, and it has evolved into this. This is the north, and it represents the past and the medium fragmentation going on in the near future, and then this is the more recent history of it. So there's this, it's kind of cool to use spatial studies to make some inferences about time and the processes. The other implication is another obvious one, is that earlier, we've had that range contraction of bears in the 70s, which was generally quit, but I think that we might have, we need to be aware, we have this fragmentation occurring now of small isolated populations, and you have the small population paradigm in some of those populations, and if we have elevated mortality, some of those might be extirpated along the way, unless, and that's the big lesson, we actually have effective management to erase those yellow lines. And just as an example here, the mountain caribou in British Columbia are highly fragmented into it. They look like a metapopulation, but they are not a metapopulation. Van Orten group, they found all this. These are the mountain caribou population sizes across western or eastern British Columbia, and there's no movement between any of those populations. So that's not acting as a metapopulation. There's not slow recolonization or whatever. They just think it's on its way. I'm not suggesting that bears are on their way to extinction. I'm just saying you gotta watch out. Even though it looks structurally like a metapopulation, there's so little female movement in the grizzly bear one, those small populations are at risk. But we're going to go through that in a minute here. So I'm going to go into my real talk. So once we've sort of documented this fragmentation, we realized it would be nice to spend some time going out and try to solve it and erase those yellow lines, basically. So this is the thing you've seen. This is the second half of that. And I'm also just going to throw a little climate change lens over this because it's sort of a theme of the conference and it's the theme of the universe in this day and age. None of us, we've had a big meeting about the effect of climate change on grizzly bears. They're a generalist species. They used to go all the way down to Mexico. We're not necessarily too worried that grizzly bears are a big threat. Climate change is a big threat to grizzly bears. What I do think, though, and you'll see a little later, and I want to say this now, is because of fragmentation and the effects of climate change on fragmentation, even though they're not affected by climate change, they represent part of the solution by trying to create connectivity. That connectivity is something we need to create to allow adaptation of climate change. And this is a bear that I work on occasionally, and it lives in a very dry environment. We just radio collared this. Chadwick was there with us. We put a radio collar on this bear in, where were we? Oh yeah, the Gobi Desert. And there are more plants per square meter in my office than there are in the Gobi Desert. It is dry. And if you drift off from your herd and you sit still very long, this is what happens to you. Nobody killed this camel, and it wasn't that Telonix radio collar either. The wolves came along and cleaned. The wolves came along and cleaned out the hump. But the researcher said this is a camel resting pose. So it's dry there, and so you want to. I'm just saying, making the point that I don't think getting dry and hot is going to take bears down in and of itself. But back in North America, bears inhabit this north-south mountain range, and even though seems like a good opportunity for them to adjust to climate change, I don't think that's entirely necessary. But like I said, climate change is predicted to exacerbate fragmentation. There's a strong link to there, and we've all been talking about creating connectivity. And if this is our current fragmentation, is climate change going to make this a little worse, probably? We're not exactly sure what's going to happen to the snow patterns and avalanche chutes and berry bushes and fish, and all those food sources are going to shift around the landscape, and it seems from a bear perspective and a climate change perspective, it would be prudent to manage for connectivity and resilience. So we started this Erase the Yellow Lines fragmentation project in 2004. I'm the Canadian. I work with the U.S. Fish and Wildlife Service. Sorry, Mike Chalet, but the U.S. Fish and Wildlife Service, not totally irrelevant. They've been working on this project for 8 years with me. Chris Savine, Wayne Caseworm. And so anyway, we were trying to find some solutions for this. And yeah, I get this long talk. I'm never sure what the next slide is, so it's a little bit of fun for me too. I let my wife jumble them up in the morning. I look up there and go, well, what was I going to say here? Oh yeah. So we're going to just look in detail at that fragmentation a little bit. Those are basically the highways, and I'm just using them. That's generally the fragmentation pattern. You can see that the yellow lines of fragmentation go pretty close to those highways. But I wanted to point out here, you got these 44 bears in the yak population, the international yak population. There's 15 to 25 or something in the cabinet population. There's 75 in the Selkirks. There's 30 and 76. Those small populations have very little female movement in them. 25% of the bears out there, 25% of that are adult females. You got 10 or 11 females out there. And if you've got no female immigration, you got a tough row to hoe on your hands there from a long-term perspective. But what's really cool is we've got these clusters of 500, 600 to 1,000 bears. Those are our core populations, and I'm trying to convince Canada and other places to not fragment those. Those are what's going to hold this together, and we're going to have to manage bears across those lines. What we're working to do, and this paper is about, is trying to get bears capable of moving across those dotted lines. And in a hopeful, dreamy sense, maybe we can link up the Yellowstone, but that's a tall order, and we'll work on that. So we used radio telemetry, and just because I've had too many doggone maps, I'm going to show you some pictures, but we use ecological habitat modeling, resource selection function modeling, which you've heard a lot about, and we're trying to identify linkage habitat. We're going to let the bears show us, those male bears that are moving, we're going to let them show us where the best habitat is, and we're going to try to inform connectivity and manage that metapopulation into being an interlinked thing. And we're not going to erase those yellow lines, but we're going to let bears move through there and stay alive. And by extension, hopefully other species. I sort of work on bears under the umbrella hypothesis. I hope they do represent other species. Here's what linkage habitat means. Let's just say you got two high quality habitat areas. You got a Sea of Humanity in the Creston Valley. There's Bonners Ferry just down here. And we're looking for low mortality risks, decent habitat that crosses through human environments, and we're going to call linkage habitat the best, safe, decent habitat to get through humanity. And we're going to work in this area right here. You'll see that. Just to show you how dangerous catching grizzly bears can be. This is just kidding. There's one of our better days last year, just to put some pictures out there. My friends were taking a picture of me doing this. You got to check the bear, make sure he's asleep. And they were sure that I would lose my right piano playing, flat picking, banjo picking hand there. But that little guy cooperated and let us go. I got to have a few bear pictures. But we take it pretty seriously. We use oxygen and treat our bears pretty well. We have pulse oximeters. It's almost like when you go into the hospital. We handle our bears, take it pretty seriously. And so in this analysis, we've colored up to about 42 bears, but we did this analysis. We had 24 bears. We get 1 to 2 locations for about 1 to 2 years. We have 35,000 data points, and we're looking for correlations between where bears were located with their GPS units and a random assessment of random points, what represents the habitat. So we're looking at, it's a very simple logistic regression. What explains where we caught bears to what's generally available? It's not very complicated. But at any rate, we tested against greenness, which you heard a little about yesterday, and ruggedness and wetness and radiation, highways, buildings, roads, canopy openness, alpine, just a bunch of different habitat variables. I'm not going to go through the modeling process. We follow all the rules. This is I put this in it for Doug Chadwick. He wanted some math equations. He can't follow these talks unless there's some good math up there. It takes about an $80 bottle of scotch to have that explained. So yeah, we can do it. It can be arranged. At any rate, we used the 34,000 bear locations. So anyway, this is another point. You're not going to see any details, but some of you guys are getting into modeling. I want to stress this, that there's a lot of models. A guy will get a handful of data and he'll run it up to the Yukon, and it's really important to test your models and validate them along the way. And me and Wayne have a rule. We don't like to extrapolate out where we can validate it with real data. So it's kind of important. I'm not going to go through. Some of these are better validation techniques. I consider this to be a little circular. You take 20% of the data and you hold it out, and you build the model with 80%. Well, it always works. You know, I've never seen it fail. But a better technique is to pull out some independent data, save some bears in an adjacent area, save some data, and ask, can my model predicting in area A, predict where these independent set of bears, how they use habitat in area B? It's a much more powerful use of data, and we spent a bit of time doing that. So here's what that data looks like. This is the Selkirk Mountains and the Purcells. We did a kind of a multi-mountain area. What it does is it doesn't allow us to get a very specific, what's that bear doing in this mountain range? Because there's no avalanche. Whoops. What are we doing there, team? I've probably shut the thing off. There's no avalanche chutes in this area, so it's one of the best bear habitats there are, but we couldn't use it as a variable. We had to use other characteristics, higher level characteristics that are more generalizable. And here's those areas we set aside. We used this as a validation area, and this is the Cabinet Mountains, and some of these bears were even transplanted over from the coast, but we wanted to ask the model we build here, predict what bears do here. It's a very nice independent test. And I'll just give you a second on the winning model. You're not seeing any of the details. It won clear hands down in an AIC test. And what's really cool about this model is it's got no negatives in it. Usually you can predict where bears go by where they avoid. And we had a lot of places with access management, and bears were just allowed to use the habitat without trying to avoid people. So we got a model that predicts what I call a positive habitat variable. These are the variables that bears are attracted to for habitat reasons. Greenness, which is one, it's a leafy green index of where deciduous foods might be. It's a very good predictor. The next best one was canopy openness. That is where there's not very many trees, light gets down in, and bear foods grow up underneath them. Also riparian areas, which are along stream sides, known as good habitat, alpine areas, and then elevation. So it ended up being, after the more and more I looked at it, as a pretty cool model because it predicted where bears are likely to go. So here's what the model looks like. This is the top third of RSF points. This is where we expect grizzly bears to be. I'm not showing you all the different colors. This is just a way to point out the good places. But at any rate, I'm not going to go through the details, but I'm just going to again stress. You can see from the graphs we had good. The closer the two lines are, the better our validation process worked. This is the validation of the independent data. In the southern Selkirks, it did much better than the model, and this one did a little worse than the model, but these were augment bears. Some of these bears were moved in for the Rockies, and we still did a pretty good job of predicting where they would go. So anyway, the hard part was to translate the habitat model into linkage zones. How do we do that? And I'm just here to have a little fun with all the least cost path analyzers. I kind of tease them. I say, you got to get out of the office and get out of the truck a little bit. I don't like the least cost path. It's too button pushy and too much GIS. So we went out and just tried to understand what our animals do, and we built some. We used what bears actually do, and it's also very intuitive. Anybody can understand it, even me. And we'll see if you like it. It's passed the scrutiny of several big-time modelers, but it's Mike Proctor's homemade linkage zone identification method. But it has three criteria. It's pretty simple. We took the places in the valley bottoms with the highest RSF scores per valley segment. You know, here's a valley segment. There's 20 highway segments, and we want to know what's the best here for this bear, not what's the best in the whole study area, but what's best in this segment. So we used one standard deviation above the mean and took the best RSF scores because we know bears come down and visit those valleys, even though they don't cross over. It's a very important habitat. We also used then, and this is a little more complicated, the best RSF score in adjacent to the valley bottom. You don't want a bridge to nowhere. You want to know where's the good habitat behind the highway, and then where's the good habitat at the highway. And we tried to bring those two ideas together. And then we know that running a linkage zone right through a bunch of humans is not a good idea. So we also just did an overlay where there's a lot of humans, and what we did is pick places where all three of these things met. And I'm not going to spend much time, but I'll show you what that means. Here's a little highway segment, and the blue is the highest RSF scores in the valley bottom along that highway. And then we extrapolated that all the way across our study area, across maybe about 9 or 10 highways. And all the little blue spots are where the RSF scores good in the valley. Pretty simple. Now we're going to look at RSF scores adjacent to the valley, and this is my worst slide. It doesn't really make a lot of sense. You're going to have to trust me. The lighter color is where it's good in the backcountry on each side, and this is where we can go in and look and find that the adjacent core habitat is best in the valley. It's a little complicated. But those green areas are where those link up. Those are the areas in the valley where it has the best adjacent grizzly bear habitat. And then the other one is a little simpler. We just took and buffered our places and found the places along the highways where there were not many humans. Pretty simple. And then what we did, we defined a linkage zone as places where all three of those overlapped. We had to have low humans, good habitat in the valley, and good adjacent habitat. And we came up with a map across Highway 200, 56, 95, and up into Canada. And we got all these green blobs are the linkage areas across that whole area, a pretty large area, across about 8 or 9 highways. And what we had to do, and this is another important, we went to a little bit extra effort to validate our linkage zones. Are they any good? So that's a little hard to do. We didn't really have a way to do that. So there's 2 areas where somebody else did a similar thing using local bears and a completely different method. I was sort of part of one of them. But Clayton Apps wrote a paper in 2004, I guess we all did, up here when we started this DNA survey stuff, and we predicted linkage zone stuff up in Highway 1, the Trans-Canada, and there's the red circle is where Clayton Apps' paper with local bears and using genetic data only predicted linkage zones. So we did our model 250 kilometers away, ran it up there, and it picked exactly the same spot. That made us feel good. So then we went over to Clayton Apps. He's always a little bit ahead of me, the guy. Trying to put a sea anchor on his leg so he won't do everything before I get there. But at any rate, he also did the same thing with independent bears and independent area over in the Rockies along Highway 3. You've all seen his paper. John Weaver was part of that. It's very good work. And his work picked out 3 linkage zones, and ours got nailed the same exact 3. Well, this says 2 things. One is we did a good job, and 2 is it ain't that hard to pick out that linkage zone. And we both got it. So we're starting to believe in this stuff. You gotta kind of convince yourself. So, just as you can see that, here's our model area, here's our model link validation areas, and our linkage zones validation areas. So we're feeling pretty good about those linkage zones. You don't want a bridge to nowhere, and I'm not going to go through any details. I'll just zero into this. We took that habitat model and kind of smoothed it and clustered it and tried to predict the areas that are high quality behind the linkage zones. And here's a good example, Highway 2 down by Troy and Libby. You've got a core area in the cabinets that's linked up to another core area by a linkage zone. That's a perfect little example of having your linkage zone linked to high quality barrier areas. Pretty easy stuff. It was really fun to see the link stuff yesterday because what really needs to be done is with 5 or 10 species, and we mill them all together and we see where we should go in the landscape. This is the same map in color. So we've taken these, I'm zeroing in on the end here, team. We've taken these linkage zones in Canada, where we started a lot of this stuff, and have written a linkage zone management plan to give to society. Say, listen, here's what we think you should do in these linkage zones for bears and wildlife to manage them in a wildlife-friendly way to get animals moving along there. And I'm just going to highlight one of those aspects in that report, and you can find that on our website and stuff. This is kind of a cool, we decided to actually try to help implement the solutions rather than just kind of describe them. So we've got sort of a lot of this work has stimulated an NGO network, the Nature Trust of BC, Vital Ground, which a few representatives in the room, and Nature Conservancy Canada, Harvey Lock, and the Y2Y, and other forest legacy programs to try to help purchase some lands in these linkage zones to help, help this whole process. It's been pretty neat. We've been working on it for several years. And I'll just show, we've purchased some land in three of these linkage zones right now. We're working through it. I'll show you just one real quick example. This is this Duck Lake linkage zone, and you can see this female bear lives on this side of the valley, and she decided to cross over to the other valley and check out the guys on the other side. She was sort of a young female. She's a little teenager. So she walked all the way up to my linkage zone. We caught her after we identified these, crossed over, went to the other side, and said, oh, you know what, I don't like it over here. And then she went back, went all the way back to our little linkage zone and came back. It gave us some confidence that the bears are making some decisions to actually use these places. It's not just random. We see this over and over and over, that the bears are using these places. And so in this area, this is the same linkage zone. We've itemized some areas. We can't buy all the land, but we can buy strategic pieces to kind of help. And this is going to be an example of our multiple species approach here. We've got a property that we're just about to buy right here. I call it the frog bear property. British Columbia's threatened leopard frog colony is right here, and the bears and the frogs use this little piece of property to get back and forth. And there's, I put a camera up right here at this linkage zone, and I got almost, it looked like the talk we showed. There's almost every mammal in the world running across that using that little linkage zone to link up. And this is between the Purcell and the Selkirk Mountains, a very important place in all this. So we're really trying to link this up to multiple species. That's where I think this sort of shines. We're using the sexiness of bears to buy a frog property. Pretty darn cool. And out of deference, we call it the frog bear property. Don't be calling it the bear frog property. So there's another little place we've been buying some property along here. It just shows you how we're actually, some are under negotiation. We spent about $2 million. There's more in the hopper. We're actually doing it. We also get out on the ground and try to retrain. We've got a retraining program for humans. We've got you can catch a person with a six-pack of beer. You can't believe how easily it is to catch them. We send them to a retraining camp. We got down and it's hidden somewhere in North Idaho. And we retrain them, put them back out on the land. You release them. They usually stay in the same home range. You know, sometimes they shift wives. It gets a little chaotic, but it does work. But anyway, the bear who did this is probably dead. You know, he's not going to live through this. This is another bear that one of our research bears wandered into a yard. But what we do is you guys do this is old hat in Montana, but it's kind of fun. We've stolen some of your techniques to manage. I'm trying to train that bear. I'm trying to train that guy who attracted the bear, and I'm training our CO. It's kind of quite the program to do these non-lethal programs and basically getting them to tolerate wildlife. But it's not just bears. We got a lot of elk problems and stuff. So here's my other big video. I'm pretty proud of this. I want you guys to stand back. There you go. You can get a replay. We're up here in our Mad Max uniform. That's me in the middle there. You got to get away from the bear. Sometimes you open that door, you know, he comes right at you. And so we're up 15, 20 feet high. This guy's got a live round, and our CMP guy said, if that bear attacks us, I just don't shoot him until we bear spray him for a little bit and see. And he's going, you're crazy, man. I'm not standing here. But at any rate, it's good fun. And I tell them, you still get to shoot the bears, and you know they live. So this bear, it worked really well. 2 or 3 years later, he still's never come back to any place. We keep him in jail overnight. And that's pretty much it. This is our website, the Transborder Grizzly Bear Project. And you can see a lot of this stuff on the U.S. Fish and Wildlife Service. It's a catchy name. I had a contest to get that name, transbordergrizzlybearproject.ca. And we have all our stuff on there. And there it is. You can see these results and see what we're doing. And we're continuing to work for at least another 5 years. And I'm just going to hit real quick just to touch your memory on those themes. You know, the meta-analysis. All this stuff is a accumulation of a meta-analysis, a really powerful tool. And the cooperation it took to understand all that stuff. And to apply the solutions through the NGO contacts and the science community. It's a really neat story of science and government and NGOs getting together. Although Michael Soule was correct, the government's hardly involved in any of this in Canada, except in the U.S. You're way better. You guys are involved in the research and the solutions, but it's basically a homespun solution. We're just getting it done. And science is a very powerful action agent, and I think that's about it. Thanks.
Okay, we're running a little late, but we can't let Michael off the hook too easily. So if there's a couple questions, come on down to the middle there, and if you could just use the microphone, that would be great.
Good.
So with buying up these linkage zones, what actions are you implementing on the land? Like, are you trying to build underpasses? Are you just trying to preserve some pristine areas for the bears so that they feel comfortable getting closer to the road? Or?
You know, underpasses are a solution we're working towards. That's never going to happen until they rebuild that highway. So we give this data to the ministries, and someday when they want to twin that highway or upgrade it, we hope that they will put in some overpasses. We also have, basically we're trying to minimize remember how the settlement quantity really affected female movement. We're trying to limit the human population. That's why we don't have to buy up all the land. We're trying to buy up some of the land and turn it into conservation lands or conservation easements to minimize the human population, number one. Number two is we go to the forest companies, if it's public land, and we tell them where it is, and they try to manage their forest. You don't have to stop logging, but manage their forest practices in a bear-friendly manner. A lot of times that means closing the roads in those linkage zones after, so the bears feel comfortable. And then we have that human retraining program I was joking about. We do that, and so basically trying to get, we got to coexist with these animals, and it's amazing how it can be done. But you really got to teach the bears and the people how to do it. So. John.
As usual, wonderful work. And I was just wondering if you have any evidence yet of bears actually, with all this reconnection, actually contributing genes to the adjacent population.
Yeah, it's an excellent question. We do. We are doing our next line of papers is we're running all our genetic samples and up to 21 loci. So we get really perfect parent-offspring relationship. When I say we, I mean WGI. And that allows us to monitor reproduction after an animal moves. So it's one thing to have an animal move, but it's another to demonstrate functional connectivity, which is sex across the highway. So we've done a pilot study on that, and it's really cool. We got a male who runs across and has a little girlfriend north of the highway, and we've got brothers and sisters who are on opposite sides in different places. And we've also, the southern Selkirk population is completely isolated. It has been for several generations. The genetic signal is very strong, but we've got two new migrants, one female and one male in there. So we're documenting. Not only are we applying the solution, but we're going to keep monitoring and document the solution. So we'll have a paper out about that real soon. We're just in the analysis. Pretty neat. So thanks for that question.
If males are crossing the highway, why isn't that adequate to spread genes across?
It's not. It does a really good job, and that's why I didn't talk about it. I was hoping you were here for that, guys. There's a difference between genetic connectivity and demographic connectivity. You got small populations of 40 or 20 bears in it, and there's no females running in there. You can run genes and males in there forever, but will that really ever help that population be self-sustaining? No. You'll just run in yet another eager sperm packet. But what you need in there is if you can get females to move in there, then you have a chance to, that's the reproductive engine of the population. That can, they'll be able to grow more kids. So that's a demographic problem. Genetic problem, we've seen the genetics of the Kodiak bear is like 30% of the genetic diversity of the rest of the continent of bears, and they are doing fine. We're not seeing much evidence that genetics is a problem for bears. But we do know that humans kill most of the bears, and in the short term, that demographics is their threat. So we're trying to connect. I appreciate this question because I didn't make it so clear. We're trying to get the demographic connectivity on there so stochastic mortality doesn't take that female population down too low. Does that make sense?
Yeah,
it's
does.
because of those populations are small. This is not an issue in the Rocky Mountains where there's 1,000 bears on each side of the highway. That's not. There's no demographic issue there. There's no genetic issue there. There's a long-term fragmentation problem. But it's those series of clustered small populations that are at real threat because first of all they need a large population, and then we need to be able to get some females in there. In Canada, there's like 5 or 6 females in the yak population. We lost 3 of them one year. What do you got? You got 2 females in the Canadian population. So if we don't get some more females in there, you got nothing. So that's the issue. And I'm really glad you point that out because demographic connectivity is really the issue. And that's what was fun about this, and you needed to study individual, you had to have an individual-based study to find that, right?
Okay, thank you, Michael.